If you were born after 1970, welcome to the modern environmental movement. Make sure you've packed your 3 R's, a reusable water bottle, and some, hopefully sustainably sourced, Earth Day meal. By the way, happy early 53rd birthday, Earth Day. To this day, upwards of 75% of Americans worry about the general environment a great deal or fairly often, and 85% feel that plastic waste is a serious and concerning problem that requires immediate political action, whatever that means. But it's safe to say that Americans are aware and angry about our plastic problem. But how bad is plastic waste accumulation really? Well, in 2018 alone, the United States generated 35,680 tons of plastic. That's equivalent to 9,000 African elephants. Hello! And if you want to visualize that much plastic, imagine filling up an Olympic-sized swimming pool 67 times. Compare this to 1960, when municipal plastic waste for the entirety of the United States could only fill 3/4 of one pool. Quite a change. But I recycle, you're saying to yourself. Did you know that only 1 in 4 plastic items you recycle are actually recycled, and that the rest are landfilled? You might as well throw your plastic into the garbage unless you land heads twice or draw a heart from a deck of cards. Don't feel too bad though, there are lots of reasons traditional plastic recycling isn't working. Those plastic lids at Starbucks and the recyclable trays from restaurants are not, and never have been, recycled. Sticky, toxic, contaminated plastics, straight to the landfill. And don't even think about the insanely expensive and environmentally damaging chemical recycling. So what do? You want to do the right thing and recycle your plastic, but know that it's not really going where you want. The answer? Biodegradation. You're probably familiar with the idea of composting. Throw your banana peels and eggshells into some fancy dirt and boom, you saved your food from waste in the landfill. But what about cardboard or plastic waste? Well, some low-key, simple cardboards can biodegrade naturally in a few weeks, but most cardboards are corrugated or coated, both of which extend a box's decomposition time. And plastic-coated cardboards are the worst. You might as well just use plastic. But why is plastic so bad? And why can't plastic decompose like organic waste or cardboard? It's all to do with chemistry. The bonds that hold plastic together are foreign to the microorganisms responsible for breaking down organic matter in cardboard. We made plastic, and the enzymes, those are proteins that speed up chemical reactions, found in nature can't unmake it back to its original parts. At least we thought they couldn't. Until recently, it was thought that only archaic bacteria living in harsh conditions were capable of producing enzymes that could break down PET. But in 2023, scientists discovered a new set of enzymes that are much closer to home. Imagine you're a bee. You're buzzing happily, collecting nectar and spreading pollen before coming back to your hive to find destruction. Invaders have eaten through the wax holding your colony together and are turning from white pupa into full-grown gray wax moths. The greater wax moth, scientific name Galleria melanella, lives in dead or dying beehives and primarily consumes beeswax, hence their name. Surprisingly, it's within the saliva of these worms where new plastic-degrading enzymes have been discovered. Hexamerins are a specific class of enzymes, previously thought to primarily function as protein storage. However, many other functions can be carried out by hexamerins, including plastic biodegradation. The 4 enzymes of interest discovered in waxworms are Sibelius and Demetra, which work together as a team, Ceres, which is a metal-binding hexamerin, and Cora, which is the main protein of interest for our research, and the most abundant. All 4 enzymes are responsible for oxidizing and breaking down PET. So we know that waxworm saliva has 4 key enzymes responsible for degrading PET in naturally occurring biological system. Where do we go from here? Well, one detail still remains unclear, and it might hold the key for how we can harness the discovery of these proteins on a larger scale. How does this newly discovered Cora enzyme degrade plastic really? One possible theory relies on the free tryptophan binding pocket found within the Cora enzyme. But hold it. What's a free tryptophan, and what on earth is a binding pocket? It's important to understand that enzymes aren't just simple, small linear molecules like glucose sugars or triglyceride fats. Enzymes are protein chains, often hundreds or thousands of amino acids, those are building blocks, long. And they don't stay in a chain long before folding up into complex structures with very specific shapes. Imagine the key to your house. It opens your door specifically. Now imagine if the shape of your key was suddenly changed. It would no longer be able to unlock or bind to your door. Enzymes are the key, and what they bind to is the lock. There's a specific association between them. In the case of the Cora enzyme, the unique way it folds creates a physical 3D pocket to which substrates, small things, can bind. One of the things that can bind is a free tryptophan molecule, which is one of the amino acids we were talking about earlier. When this tryptophan binds, it's possible that the shape of the enzyme is modified or stretched, allowing for its biodegrading properties. To see if this is true, we ask one simple question. Is the sequence of DNA that codes for the binding pocket highly conserved among individuals? Some genes, like tumor suppressing genes, are important to long, healthy life and are therefore highly conserved between individuals. That is, a mutation in an important gene would likely not be passed on to the next generation. On the other hand, less important genes, like those encoding hair color or earlobe attachment, are likely to be highly variated between individuals. If core's binding pocket is important, it'll be highly conserved between individual waxworms. But how do we know if a gene is conserved between individuals? Well, to compare DNA sequences of waxworms, we're going to need to first isolate DNA from wild and commercial waxworms, amplify the sections of DNA we care about, confirm that those amplification works, sequence the DNA collected, and finally align our sequences to determine overall variation. Step 1. Isolate the DNA. We started with samples of both wild waxworms, which were harvested from apiaries, and commercial waxworms from industrial sources. We are interested in the difference between these groups because we believe that commercial waxworms will have less variation in general, and want to see if this is true. To isolate our DNA, we crushed the worms, don't worry, they were already dead, and washed the remaining liquid many times until we were sure we only had DNA. Step 2. Amplify the DNA. Even though we just isolated DNA, a couple molecules isn't enough to do all of the other experiments. We can't just magically tell the DNA to duplicate, but we can use our understanding of how DNA replicates naturally to artificially obtain the same result. We used a process called PCR to amplify exons 3 and 5 of the core gene, which are responsible for encoding the binding pocket. Step 3. Confirm our DNA. In order to double check that we amplified the right regions of DNA, we ran a gel electrophoresis on our products. A positive result shows a DNA product with fluorescence under a UV light, while a negative result is blank. Step 4. Find our sequences. If PCR worked correctly and we accurately amplified the right exons, the next step is to determine our sequences using Sanger sequencing. Sequencing is a technique that produces the exact nucleotide sequence of a DNA molecule. Step 5. Aligning our sequences. Finally, we can align the resulting DNA sequences, which may have slight variations at the beginning or end, so that we can better see nucleotide differences. Some of these differences may cause a mutation in the protein which causes it to not work, while others have no effect at all. More importantly, we can see the overall similarity or dissimilarity between individual samples. So, what were our results? Before we unpack the exclusive conclusion on how variated exons 3 and 5 of the core gene are, let's do a quick recap of how we got here. We crushed our worms and isolated our DNA. Mine had a concentration of 18.9 nanograms per microliter. That's about half as how much sugar is in a can of soda. Then we amplified the DNA and confirmed our products using gel electrophoresis. Mine only worked for exon 3, unfortunately. Finally, we sequenced our DNA and compared our results as a class. Here's my worms, worm number 250, DNA sequence for exon 3. How cool! There's just one more step. Get everybody's sequences and line them up so we can see the specific differences between them. And here they are. Ta-da! Well, that's super underwhelming. They're all the same. What gives? Well, even though these results look boring on the surface, zooming in gives us some big insights. The most important thing is that these sequences aren't variated at all. An individual with a serious mutation here probably isn't making it to the next generation. Next, when there is a difference, it's a single nucleotide, and it likely doesn't even have an effect on the protein produced. Finally, take a look here on the left-hand side. Sequence alignment tools put the most similar sequence at the top and the least similar at the bottom. From top to bottom, the order is commercial, commercial, wild, commercial, commercial, wild, commercial, 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 wild, wild, commercial, wild, wild. There's no order to that at all. The commercial and wild worms have the same sequences. This gene is super important. Great. We know that the binding pocket is really important for core's plastic biodegrading abilities. What now? Well, as with any scientific discovery, our updated understanding triggers dozens of new questions. What is the free tryptophan actually doing in the plastic biodegradation process? Could we genetically modify the core enzyme to act even more efficiently? Can we get bacteria to produce this enzyme in an industrial setting to make a new form of plastic recycling? Investigating this last question in particular might be the answer to America's plastic problem. What if biodegradable plastic was every plastic? And instead of tossing contaminated plastics, we got core to break them down instead. What if 100% of what you tried to recycle responsibly actually made its way back into the environment? What if the environmental movement born in the 70s didn't die in the 90s and resurfaced today? Well, I for one believe our world could be a much greener, more sustainable place if we simply use science to worm our way out of this one.